The main types of allergic responses to food antigens are type 1 and 4 hypersensitivity reactions, which mediate the acute and delayed phases of allergic responses respectively. The acute phase occurs between 5 minutes and 2 hours after allergen exposure, whereas the delayed phase occurs about 2 to 3 days after. A response may be mediated by one of these phases only, or both together. So both begin with allergen uptake and processing by a type of immune cell called major histocompatibility complex class 2 or MHC2 cells, which are specialized for antigen processing. So the MHC2 cells break down and process the allergen into little fragments and then present these on their surface. The allergen fragments are detected by T cells, specifically the T helper cells. Which are involved in many immune signaling processes. So type 1. Is where IgE is activated, a type of antibody specialized for allergic and parasitic responses. So following the T cell stimulation, activation occurs via a cytokine or signaling molecule called interleukin 10 or IL-10, which turns the T cell into a T helper 2, Th2 type cell. These cells release other cytokines, IL-4 and IL-5, which signal to B cells. B cells are another common class of immune cells specialized for antibody production. The IL-4 ensures that the stimulated antibody production will be of the IgE type. These antibodies have a high affinity for their antigens, so a small number of allergen molecules is sufficient to trigger a response. The IgE antibodies cross-link with each other, and the IgE antigen receptor binds to the allergen, whereas the Fc, the constant region receptor, binds to another type of immune cell called mast cell. These cells are specialized for allergic responses and release inflammatory mediators such as histamine to cause constriction and mucus secretion in airways. Increased dilation and permeability of blood vessels, and also increased peristalsis or muscle contraction in the GI tract. No symptoms occur upon first exposure, while the B cells mature and begin initial IgE production, but upon subsequent exposure, a greater body response is mounted due to immune memory caused by the B cells, and mast cells are then activated and symptoms occur. The common example of an IgE-mediated hypersensitivity is peanut allergy. Okay. Type 4. Is the cell-mediated delayed type response. Mediated primarily by the action of T cells. The T cells that recognize the MHC class II are activated by the cytokine IL-12 to produce T helper 1 cells. These cells then release interferon gamma and tumor necrosis factor alpha. These are pro-inflammatory molecules that recruit immune cells such as macrophages and neutrophils to the site of allergen exposure to aid clearance. And this results in symptoms such as edema, i.e. swelling. This action is but in this case, symptoms can occur with initial exposure. However, the memory response is highly vigorous, so each subsequent exposure can be dangerous. An example of a type 4 or non-IgE mediated hypersensitivity may be celiac disease. Which is a response to gluten, though there is debate as to whether celiac disease is an allergy or an autoimmune disorder.